Not yet.
It says recording pending. Okay,
Like it is.
the recording started. Without further ado, Justin, you have the floor.
Okay, thank you for the introduction, and thank you all for coming to my talk today. So I'll be talking about my project on developing a brain-computer interface based on visual imagery that I worked on this summer with my mentor Yvonne and the rest of the BCI and audio and acoustic research teams here at Microsoft. So first off, starting with a little bit of background into this research area, and I'd like to ask the question first of what exactly is a brain-computer interface? And in its simplest terms, a BCI is a technology to facilitate communication between the brain and an external device. And importantly, this communication does not depend on any inputs from peripheral nerves or muscles. It's just a direct communication between the brain and the device. So a basic framework of this experiment, you might have a person engaged in some sort of task where they're, maybe imagining interacting with some object on a computer screen. First off, you're acquiring their neural data. That data is being processed and then transmitted over for classification and translating that code from like a neural stimulus to a control code to control something on the device. So the first step in any BCI is determining how exactly you're going to measure the neural signal. And there's a few different methods, each with their own pros and cons, but one major consideration is the spatiotemporal resolutions of each of these devices. So I'll go through a few of them, starting off with EEG, or electroencephalography. So EEG is like electrodes placed along the surface of the scalp that can pick up the small electrical potentials that occurs when neurons fire. So EEG is perhaps the most common, most well-used device for BCI applications because of its relatively inexpensive starting cost. It's pretty portable, like you can have just a cap that you slide on easily or electrodes embedded into some other device like headphones, something like that. It's got a pretty high temporal resolution, but it has a low spatial resolution. So because you're trying to record the signals through the skull, through the scalp, it could be a little bit difficult to localize where exactly those brain signals are coming from. So next up in the non-invasive side is fMRI. So this would be functional magnetic resonance imaging. This is actually what I'm using in my PhD research right now. So fMRI has a much higher spatial resolution than EEG. You can get like small, like almost millimeter size called voxels or brain area, which might have, a few thousand neurons in there. So it's better spatial resolution, but it has a very low temporal resolution. So you're only able to capture an image like every second or two. Another issue with fMRI is it has a pretty big delay because it's an indirect measure of brain activity. So like with EEG, you're measuring the electrical potentials of the neurons directly, but with the fMRI, you're actually measuring changes in oxygenated blood flow into an area of the brain. So the idea here is that once a region of the brain becomes activated, the body has to feed that area with additional oxygenated blood. So fMRI is actually measuring that increased blood flow in an area sort of as a proxy for brain activity. So because of this, you have to deal with an issue known as the hemodynamic delay, which it takes about like 4 to 6 seconds for you to be able to determine that, blood flow is increased in that area. So it can be a little bit difficult timing, like when an action corresponds to an activity in the brain. So moving on to the more invasive techniques, one common one is electrocorticography, or ECOG. So this is pretty similar to EEG in that it uses electrodes, except now they're placed along the surface of the brain. So this has a much higher spatial resolution and still taking advantage of the high temporal resolution of EEG. However, of course, it does require an invasive surgery, which has its own inherent risk with like infection, things like that. And also, these electrodes can't be left in for a very long period of time because, over time, as the body's reacting to this foreign object in the brain, you might have like scar tissue buildup, things like that, that can cause the signals to degrade over time. Even Farther in the invasive side, there's like local field potentials or spike electrodes. So these would be electrodes placed into the cortex that could read like a single neuron or like clusters of neurons. So it has a very high spatial and temporal resolution. However, once again, it requires invasive surgery. The signals can degrade over time. It's got a high cost and a high power consumption and latency issues. So next, I'd like to talk about some of the common BCI paradigms used in BCI applications. So these paradigms can fall really into two different camps of evoked potentials and non-evoked potentials. So an evoked potential would be, providing a participant with some sort of external stimulus, and you're measuring the brain's response to that stimulus. Where a non-evoked potential would be, participants engaged in some sort of imagery, so like an endogenous stimulus, and you're just measuring their brain activity during that imagination. So in the evoked potential, possibly the most well-studied and well-known paradigm is the visual P300. So this is a common event-related potential that appears as a reaction to a presented stimulus. So one application of this that you might have seen before is the P300 speller. So how these devices work is you would have a subject be able to spell out like a word or a phrase just by looking at one of the letters at a time on this keyboard. So the different rows and the different columns are going to be highlighted at different times. And based on what the participant is looking at and the changing intensity levels of that letter flickering on and off, you can judge, when the letter highlights and when the ERP response happens in the brain, which letter that they're looking at. Another kind of similar technique is the steady state visual evoked potential. So how this would kind of look like is a person would be presented with this array of flashing targets or like flashing LEDs. Each of these targets will be flashing at a different frequency. And based on which target that they're looking at, you can match the frequency components in their EEG to the flickering frequency of the target and identify which one they're attending to. Some less studied evoked potentials could be like, measuring a person's response to an auditory stimuli or to like a somatosensory or a tactile stimulation. And then moving on to the more like non-evoked side of things. So this would be, mostly imagery-based paradigms, and by far the most studied one is motor imagery paradigms. So for example, of a motor imagery paradigm like sensory motor rhythms, this could be the imagined movements of large body parts. So for example, you might be imagining opening and closing your right versus your left hand. There's also imagined body kinematics, which is like the imagined movement of a single body part in a multi-dimensional space. So this is actually a technique that I use quite a bit in some of my past research. So an application of this might be like if you're trying to have a BCI to control a computer cursor, like on a screen, you can have a participant imagine they're moving their right hand as if they're using a computer mouse to control the movement directions. Some less studied non-evoked potentials could be like visual imagery. So this would be like a conceptual reconstruction of a perceptual experience. So like directly imagining an object that you're trying to interact with. There's like speech imagery, which is like the imagery of speech production. And there's different categories of mental effort. So this could be things like, imagining rotating an object in a three-dimensional space or mentally performing math calculations, something like that. So to give a little bit of background into this project that I was working on this summer, my main goal was to create a BCI using some kind of non-evoked potential. And we wanted to kind of steer clear of motor imagery. So for that reason, I selected visual imagery as the paradigm that I was going to use throughout my experiment. And one reason for this is, we wanted to do something a little bit different that hasn't been studied as much. And also visual imagery provides like a more intuitive control for a BCI. So say, for example, you want a BCI to control different applications in your house, like maybe turning a lamp on or off. With visual imagery, you can imagine just that lamp directly and interact with it. Whereas with something like motor imagery, you'd have to remember a map between your right hand might interact with that lamp. So next, I'd like to just give a little bit of background into some research in the visual imagery BCI field. So one study by Natalia Kosmyna from 2018. In this experiment, she was trying to perform classification of visual observation and visual imagery periods between two object classes of either a flower or a hammer. So you can see her task stimulus down here. The participants would first be shown like a image cue of, say, like in this case, the hammer. Would appear on screen for 4 seconds, and they would just be asked to, attend to that stimulus, and then it would disappear for 4 seconds, and that would be like an imagery period where they tried to, imagine the stimulus that they just saw, and then there would be a rest period. So for her results, particularly for like the observation periods when the picture was actually on screen, she had decent classification accuracy. However, when the image was taken away and the participants were asked to just imagine it, and she was only able to find around 52% decoding accuracy, so right around chance level. In another study by Lee et al. In 2019 and repeated in 2020, in this study, they were trying to perform classification of visual and speech imagery, and they had 13 classes of like common words for patient communication. So these could be things like bathroom, water, ambulance, or more abstract concepts like help or yes. So for the visual imagery classifier, when they first did this in 2019, they were able to get around like 27% accuracy, which doesn't sound great, but with 13 classes and chance level being around 8%, like there's still something there. You're able to classify between the categories. And then when they repeated this a year later with a larger cohort of participants and some different processing techniques, they were able to increase this classification pretty remarkably up to around 40%. So the next thing I'd like to talk a little bit about is some of the pre-processing steps that you would go through with working in a EEG BCI. So starting out, you have this steady stream of raw EEG data come in. One of the first things that you might do is perform a channel selection. So perhaps at the beginning of a study, you might work with like all the channels in the cap, but sometimes depending on what the task that you're doing, it might be beneficial to narrow down those channels to a particular region of interest. So for example, if you're doing a motor task where you have your participants moving their right versus their left hand, you might be interested more in like those C electrodes directly over the left and right primary motor cortex. Or if they're performing like a visual task, you might be more interested in the O electrodes on the back of the head directly over the visual cortex. Next step you would probably do is apply a filter to the data. Usually at this stage, it would mostly be like eliminating any low or high frequency noise from the signal that you're not really interested in. So EEG is usually grouped together in these bands of brain activity. Each of these bands kind of correspond to a particular state of the brain. So for example, like delta waves are usually seen in like a deep sleep, like mu and beta waves are seen during like motor movements and executions and imagery. Alpha waves usually carry information about like subjects' attention. So if you're trying to see like if they're attending to a task, like alpha waves usually carry that as an example. Some other filters that you might apply is just to remove noise like power line noise. Another step that you might want to take is perform some sort of artifact removal. So EEG can be pretty prone to artifacts from like motion, like eye movement, from like eye blinking or muscle movement from things like chewing. So the picture up above kind of demonstrates what an eye blink would look like, particularly on those frontal channels. If you're doing any sort of analysis that is taking advantage of those frontal channels, it might be beneficial to remove those eye blinks. Also be a good idea to epoch the data. So this would be like dividing the data into smaller windows. So this would kind of depend on, like the task period, if you want to divide the data into like when the task starts and when it ends, or maybe even break that into like smaller categories that you can process your data and classify it based off of. And then finally would be like feature extraction. So where you would extract the time or frequency components from the signal that you'd like to use as features during your classification. So like, for example, maybe before you started with a broad filter of EEG, like filtering from like 1 to 50 hertz, but now you want to narrow it down to just use like the alpha band of activity for feature selection. So you might use like a FFT to pull out just that like 8 to 15 hertz range. So next I'll talk a little bit about my experimental methods and the protocol that I used here. So the objective of my project was to create a brain-computer interface using a visual imagery control paradigm. And the recording device that I used for this experiment was the Brain Products ActiCap Express Twist. So this cap featured 32 channels of dry electrodes with a 500 hertz sample rate. So usually with EEG applications, you'd use like a wet electrode system. So this would have a small amount of conductive gel placed underneath each electrode to improve the signal quality of it. So unfortunately with the dry electrodes, due to me having to work at home alone, it's a lot easier to put on a dry electrode cap. However, they are a little bit more prone to noise and you're a little bit limited by how much data you can collect. Because if you can imagine like 32 of these little spikes pressed against your head, it's kind of difficult to wear that for a prolonged period of time. So I can only maybe wear it for around like 30 minutes before I start getting a pretty bad headache. So for like the overall framework of my project here, the programs that I use are OpenVive, Lab Streaming Layer, PsychoPy, and Python. So for this, I have like a participant, engaged in visual imagery where they're, imagining an object that they're trying to interact with. The visual imagery EEG is recorded through OpenVive, which transmits the signal via Lab Streaming Layer into a main BCI processing script. At the same time, I have PsychoPy running the stimulus script. It's controlling, when the stimulus is presented for the subject and, when the feedback is presented. And it's also sending the stimulus markers to that main processing script just to tell it, when the image appears on screen, when it goes off and everything. So this main processing script is just taking in all that data and labeling it appropriately and sending it over for online analysis, which carries through all the data preprocessing, the feature extraction, and the classification. And that classification result is sent back to the PsychoPy script to display the feedback. Now, most of the experiments that you'll see coming up are going to be like offline analysis. So the only difference here is that we're not actually giving a feedback to the PsychoPy script. It's just this main processing script just saving all the data for offline analysis. So for the first experiment that I carried out, really what we wanted to do was just recreate the protocol from Natalia's study that we looked at initially, mainly just to make sure that our processing pipeline was working as intended. We were getting the results that we were expecting. So just as a reminder, this paradigm had first a fixation cross appear for a period of 1 second, a blank screen for 2 seconds, an image would pop up for 4 seconds, and the participant would be asked to just attend to the stimulus and focus on the features of it. We had 2 different classes of images of a flower and a hammer. So it was always the same flower, always the same hammer throughout the experiment, just in a random order. So once they focused on it for 4 seconds, the image would disappear and they'd be asked to imagine that stimulus that they just saw. There'd be a rest period for 4 seconds, a blank screen, and that would repeat. So I recorded a single session that was divided into 4 runs, and I had 40 trials of hammer images, 40 trials of flower images, and this was about like a little under 30 minutes worth of data collection total. So moving on to the results from this experiment, the first thing that we wanted to look at was a classifier built off of the observation periods. So the objective here was to perform a 2-class classification of when they were looking at a flower versus when they were looking at a tool. So some of the pre-processing steps that I took, first, I extracted the OZ, O1, and O2 channels. So these were the ones directly over the primary visual cortex in the back of the head. I re-referenced those channels to the average of the TP9 and TP10 electrodes. So those are the 2 directly behind the ears on the mastoids. The data was filtered with a 1 to 40 hertz bandpass filter and a notch at 60 hertz to remove the power line noise. and I epoch'd the windows. So if you remember, I had around 4 seconds during the observation period. I kind of narrowed that down to a total of 3.5 seconds of data and split that up into window sizes of 1.75 seconds with a 50% overlap. So that gave us like 3 windows for each trial. And then for my features, I used the full 1 to 40 hertz power spectrum. I used a linear support vector machine classifier and cross-validated it using a leave one run out cross-validation. So for the results of this visual observation classifier, it was pretty good. We were getting around 63% accuracy, so it was relatively good at classifying whether they were looking at a flower versus a hammer. And our accuracy of 63% was pretty close to what Natalia was getting at 61%, so it looked like everything was working as intended there. So moving on to the visual imagery classifier. So the objective here was to perform a 2-class classification of when they were imagining that flower stimulus and imagining the tool stimulus. So similar processing steps of extracting the OZ, O1 and 2 channels in the back of the head, re-referencing them to the ones behind the ears, except now we're filtering the data with the larger frequency band. So with the observation periods, we use 1 to 40 hertz, but the visual imagery, it seems like the higher gamma range contains a little bit more information about imagery periods. So we bumped that filter up to 1 to 125 hertz with a notch at 60 and 120 hertz to remove power line noise in its harmonic, and use the same epoch size. And our features here were the full power spectrum in the 1 to 100 hertz range, and use the same SVM classifier to leave one run out. And as for our accuracy here, unfortunately, we were still getting around chance level accuracy. We weren't able to fully identify if they were looking at or imagining a flower or a hammer. However, this was in line with what Natalia was getting in her experiment as well. So the next thing that we looked at was an observation period versus rest classifier. So the objective here was 2-class classification of rest versus observation periods. So most of it was the same, except for we combined all of the observation trials into a single label. So this seemed to work pretty well. We were able to pretty accurately predict whether they're resting versus looking at an image. So around like 77.5%, where Natalia was getting 75. So everything was going as we were expecting there. And then we looked at an imagery versus rest classifier. So the same kind of thing, combining all of the imagery trials into a single label and just trying to classify if they're resting versus imagining a picture. And this actually worked really well. Like we were able to get 81% accuracy here, where she was getting 77%. So that was functioning as we intended as well. So this was a pretty interesting result, how we weren't able to differentiate within like the imagery period. We couldn't really tell if they were looking or thinking about a flower or a hammer, but we could tell that they were trying to perform visual imagery and when they were resting. So the next thing that I wanted to look at was to try to evaluate this performance with different image stimuli. So it was kind of interesting that we were able to differentiate between those categories during the observation period, but not during the imagery period. And one reason for this that I started to kind of notice that might help explain this was that, the flower that we were using was a pretty large white flower, and the hammer was like a smaller, darker hammer. So maybe during our observation periods, we were really classifying like a bright stimulus versus a dim stimulus, and that might be why we weren't getting any information during the imagery periods. So to test out this, I tried, adding a few more pictures of flowers throughout the experiment, keeping the same general size, but having slightly different colors or different shapes. And then for the hammers, use the same like size hammers, but maybe at different orientations and slightly different colors. So interestingly enough here, first with the visual observation classifier, doing all the same pre-processing and classification steps as before, our classification accuracy dropped down to chance level. So before we were, around 60%, now we're down to chance. So we weren't able to identify if they were looking at flower versus a hammer. And the same thing kind of with the visual imagery classifier. Again, we were still around chance level. It seemed to kind of bump up a little bit, like maybe we had a little bit better luck with the flower category, but still our overall accuracy was mostly around chance. Then for the classifier for the observation period versus rest periods, we saw still above chance accuracy. It had a slight drop here, but we were still getting around 69%, where before we were like around 75. And then for the imagery versus rest classifier, we saw a pretty big drop here. I think before our accuracy was like 81%. Now we're down to like 61.5%. So the next experiment that I tried was including different image stimuli of a face versus scene. And this was just to see if we could try something different to help boost our classification during the imagery periods. So the idea of using a face in the scene, this is a pretty common image stimulus to use in a lot of visual neuroscience applications. So there's multiple sites throughout the occipital and temporal regions in the brain that seem to respond pretty selectively to faces and scene images. So perhaps the most well-known and well-studied one is the areas in the ventral temporal cortex, the fusiform face area and the parahippocampal place area. But there's also areas throughout like different areas of the occipital regions that seem to respond more maximally to either faces or scenes. I want to be careful with saying that, it's not like the brain is really compartmentalized as like, this area is faces, this area is scenes, this area is hammers. Like, it's not exactly like that. These activity patterns are pretty dispersed throughout the visual network, but it does seem, particularly for faces and scenes, like there are some regions that are more maximally responsive to each one. So perhaps this might give us a little bit better results. And so I also study showing that face and scene images have also shown to be effective in visual imagery EEG BCIs. So it's a pretty early study from 2011 where they tried to do a 3-class classification of resting versus face versus scene imagery, and they're able to achieve around 56% classification accuracy. So pretty good above chance accuracy there. So using these face and scene stimuli, first off with the visual observation classifier, performing similar preprocessing steps and everything, our accuracy was still right around like 60% like we were seeing before. So everything stayed pretty good there. And then for our visual imagery classifier, here's where we saw the biggest benefit. So before we were right around chance level, now using like all of the electrode channels, re-referencing them to the ones behind the ears, doing the same sort of filtering, we were now getting like 64% accuracy. So we were able to differentiate when they were thinking about the person's face versus thinking about the scene pretty well. So one other thing that I tested here was getting a few extra sessions of this experimental paradigm. So I had 3 sessions total of this visual imagery classifier. I used the same preprocessing sets for all of them, using all the channels except the ones behind the ears that I were using to re-reference, doing the same 100 to 125 hertz bandpass filter, same epoch sizes, 1 to 100 hertz power spectrum for features. And so we were seeing for, first off, the face versus scene imagery from sessions 1 to 3, it looks like we were pretty consistently around 60%. What was really cool about this data is when I tried to do like a between session classification, so like training my classifier on say sessions 1 and session 2 and testing it on like a new session data that it hasn't seen, we were still able to get like 60% classification accuracy, which is pretty cool because most of the time with EEG applications, it's kind of hard to transfer data from one session to the other. Then even for the rest versus imagery periods, from sessions 1 to 3, it looks like our classification accuracy, increased a little bit, starting around like 60% up to a little above 70%. And the between subject classification accuracy here was still pretty high, around 65%. So training on 2 sessions and testing on a left out one. So the last thing that I tried here was, splitting apart that rest and imagery periods into its individual categories. So doing a three-way classification of rest versus face versus scene. So from sessions 1 to 3, it was all performing pretty consistently above chance, around 50%. And even from our between session classifier of, training on 2, testing on a 3rd, was still giving us pretty high, around 50% classification accuracy. So next thing that we tried here was an online real-time BCI application. So, having the participants, imagining one of these stimuli and then giving them feedback directly from the classification output to see how it was working. So in this experiment, the first 2 runs were like exactly from the training ones. We had the fixation cross, blank screen, an observation period for 4 seconds where it was looking at either a face or a scene image. They performed imagery for 4 seconds, a rest period for 4 seconds, and then a blank screen for 2 seconds. Then in the last 2 runs, it was a little bit different. So during the observation periods, instead of just having the 2 image categories, they could also be cued to rest during the imagery period. And then instead of a rest period at the end, they were actually provided feedback directly from the classifier's output of, what it predicted they were thinking about during the imagery period. So as for the results from this experiment, the objective here was the 3-class classification of face and scene imagery versus resting state. The pre-processing steps were all the same as before, using all the channels except the ones behind the ears, re-referencing them to those mastoid electrodes, the same higher filter band, same epoch size. Then for the training data, I used the 3 previous training sessions as before, plus the additional 2 runs from the current session to kind of initialize the classifier. And it seemed like our accuracy was still pretty high, like right above or right around where we were getting before with 50%. We just saw a slight drop, and it looked like we were getting pretty good accuracy at identifying the rest periods and identifying when they were thinking of a face. There was a slight problem with the scene images. It seemed like that was more around chance level. It was getting confused with the faces quite a bit, but it still seemed pretty consistent of, identifying visual imagery was occurring and resting was occurring. So the last thing that I want to show you here is just a demo of this real-time BCI experiment. So first 2 runs of this is like a demo of the initial training sessions. So this would be like what happened in the first 3 training runs and the first 2 runs of the experimental session. So in each run, the person would first see the fixation cross, an observation image here. So in this case, a face image. Then it would have the imagery period for 4 seconds where they're just trying to imagine that image, and then a resting period, and that would just repeat. Observation, imagery, and then rest. So the next, moving on to the real-time feedback stages. So those final 2 runs of the experiment. So in this, they're seeing the observation cue of like a face in this trial, performing imagery, like trying to remember the features of that face, and then you see the classifier's output. Like in this case, it got the prediction right. So the next trial starts, we see the fixation cross. This time we're cued to rest during the imagery period. So we're just trying to keep our mind blank during this stage. And the classifier predicts rest, so it got it right that time again. New trial starts. This time it's rest again. Resting. And then the feedback. Then a new trial starts. This time it's a scene image pops up. So we're focusing on the scene image and then trying to imagine that picture that we just saw. And then for the feedback in this case, it got it wrong. It predicted it as a face instead of a scene. So moving on to just like a conclusion and a summary of everything that we've seen. So the objective of this experiment was to create a brain-computer interface using a visual imagery control paradigm. For the offline results that we saw, we were able to achieve above chance classification accuracy when using imagery of faces and scenes. So around like 62% classification accuracy between these two. We also saw that the visual imagery was pretty stable across sessions. So we had 50% balanced accuracy of rest versus face versus scene between those 3 sessions. And so one thing I forgot to mention before was that during these 3 class classification on the offline stages here, I was using balanced accuracy instead of just the standard classification accuracy. It's because our data sets were a little bit unbalanced in this case because we had 40 trials of faces, 40 trials of scenes, but there was a rest period each trial. So there's twice as many rests as each of those categories. So this balance measure just kind of helps to create a little bit less biased classification score, where instead of just judging how many total it got right and how many total it got wrong, it finds the classification accuracy per category and then averages them together. So it helped out for here because our classifier was actually pretty good at identifying the rest periods. So if we used just standard classification accuracy, it would have looked like a lot higher score than it actually was. And then on to our online results, we saw that we were able to achieve similar performance in our real-time BCI, around 47% classification in this 3-way of rest versus faced and seen imagery. Some of the limitations of the study and potential areas for future work is that, due to limitations of, me having to work home alone, I was the only participant in the study, so definitely need more data from more people to fully validate the conclusions in this experiment. I think it would also be beneficial to try to collect some more data, perhaps from like a wet electrode cap, to improve the data quality. So like I mentioned before, with these dry electrodes, it can be a little bit more susceptible to noise. And we're also kind of limited on the amount of data that we can collect. So like we were only collecting a little less than 30 minutes of data, so perhaps getting a little bit more each session would help our accuracy a bit more. And then finally, I'd just like to thank my mentor this summer, Yvonne. Thank you so much for all the feedback and advice throughout the summer. I couldn't have asked for a better mentor. Thank you to the entire BCI team. Your feedback and all the support and help during our meetings was very valuable for getting the project to where it's at right now. Thanks, David, for helping me get all the technical side of the stuff worked out at the beginning. And thank you, Demetra, for organizing a great morale event for us. Thank you to all my fellow research interns in the audio and acoustics groups. It was great working with you all this summer and getting to know you all a little bit. And I wish you all the best. And special thanks to Natalia for meeting with me and giving me some feedback and advice on this project. And thanks to all the research intern program coordinators who organized some fun events for us this summer to kind of give us a break from some stressful work times. And thank you all for coming to my talk and listening to me. And I'd be happy to answer any questions you might have.
Thank you, Justin. Dear colleagues, the floor is open for questions. Oh, please go ahead.
Hey, Justin, great stuff. I had I guess it's an experimental question in terms of how you do the train test splits. So with the 50% overlap, you kind of have to be a little careful in terms of making sure your training data isn't actually leaking into the test data. And so were you kind of like breaking down each three or 4 second epoch into train tests and then doing the overlap, or how exactly did you handle that issue?
Yeah, so my experiment was the full experimental session was divided into 4 runs. So it allowed you to get a little break between each run. So when I was actually, testing the data, pulling out the data for training and testing, all the training data came from 3 runs. and then the testing was from a left out one. So there was no leaking of, those windows maybe overlapping in the trial. Like it was completely different trials that was being tested on.
Great. All right, thanks.
Any more questions?
I have a quick question, Justin. In the real-time demo that you showed us, you had two paradigms, and the second paradigm actually had a real-time feedback. Is that something you're planning to use in the future, or have you actually used it already for learning during the training session?
Yeah, so during the real time, the paradigm is set up where the first two runs were like the same as the training. This was just to get some additional training data to help initialize our classifier. I initially tried fully training the classifier from previous runs that it hadn't seen before and just doing fully online feedback. But without giving it a little bit of data from the current session, it didn't perform as well. It was, I think, around 39% accurate, but adding a little bit of training data from the current session boosted that up to around 48%. So I did actually have fully online BCI, and the accuracy, you can see the predictions here, it was around 47%.
Got it. So for the feedback that you're showing to the participant, what is the benefit either to the participant or to you? Are you recording the reaction to that feedback, and you may use that as an additional signal?
Yeah, at the moment right now, I wasn't doing anything with the data during the actual feedback periods. But one thing that we were kind of initially planning to do at the beginning of this was creating an adaptive BCI to where you can use the data from that feedback period and see if you see maybe an error-related potential in their brain waves during that feedback period, we could see that the classifier got it wrong, and we could try to more finely tune our classification between that. But as for right now, I wasn't using any of the feedback data.
Got it. Thank you.
Thank you.
More questions? While some people think about a potential question, Justin, one of the most interesting findings here is the independence of the classifier from session to session. We usually see a drastic difference and reduction in the accuracy when we have a classifier trained on one session and try to use it in the next session. How would you explain the fact that you can train a classifier on 2 sessions and use it on the third without substantial reduction in the accuracy?
Yeah, that's a great question. I mean, yeah, a lot of times with these, EEG applications, you really do see a pretty drastic drop between the sessions. I mean, this could be from many reasons of fatigue during the session, or even just the electrodes being at slightly different positions. So it was pretty cool that, in my data set that I had right here, I wasn't seeing any drop between that. And honestly, I'm not really sure exactly why we weren't seeing a huge drop here. I mean, definitely to make sure that there isn't a drop with visual imagery, we definitely need to try to test this out on some different participants instead of just myself, get some more data to actually validate this. But yeah.
Thank you. More questions, dear colleagues? Oh, come on, go ahead.
Hello, can you hear me?
Yes, we can hear you.
Hi, a great talk. I really enjoyed it. I was wondering if there's any thoughts about sort of analyzing the features that might be allowing for the separability in the different classes, maybe looking at the different power bands and what actual features will be contributing to the separability.
Yeah, so what I was doing right now, what I saw with a lot of the other kind of similar visual imagery BCIs, it seemed like using features from the frequency spectrum, particularly the high gamma spectrum of brain activity, seemed to carry the most relevant information from visual imagery. However, that one study that I talked about that had like, the 13 different classes that had 40% accuracy, they had a little bit more advanced feature selection. They were using like common spatial patterns. So definitely there could be some more work on this side to see if, using some different types of features could help improve our accuracy some more. Right now, just kind of starting with a little bit simpler, just frequency spectrums and seeing how that worked. But great question. Thank you.
More questions? Go ahead, Dimitra.
Okay, I'm gonna wait for my turn if someone else has a question. Justin, you in this project, you have been both the principal investigator of the study, but also the participant. And typically, what the one wants or expects is kind of conflicting with what the other side of this study, the other side of the coin, I suppose. One of the issues you mentioned is that, for example, as a private investigator, you wish you could have more data recorded, but as a participant, at times it may get a little bit painful, so you couldn't leave the device on your head for long periods at a time. Is there anything else that you may have observed from these two sides of this project regarding the stimuli, perhaps? Improvements that could come because you've been taking that experiment yourself, that study yourself as a participant?
Yeah, so that's a great question. Some of the things that I noticed about this experiment, I mean, first off, it did seem that we saw some improvements, like when we were, adding that additional data from other sessions. So it does seem like, we are a little bit limited just by the amount of data that I was able to collect from myself each session. One other thing that I noticed that I didn't really talk about in these slides, one of the things that I tried at the very start of the summer was having 3 different classes of like face images, scene images, and then having like an object category, like showing like hammers and tools and things like that. So one interesting aspect that if I had more time to kind of dive into that more, is trying to add some additional categories in here. Because even with those 3 different imagery categories, some of our results were actually looking pretty positive there. In this, I was actually able to get 2 different participants in that. Myself and my wife participated in that, and we were both around like 40 to 45% between all the 3 of those categories. So it did look like we were still able to get a pretty good above chance classifying between 3 different image categories instead of just 2 is here. And I wish I could have gotten some more data, but yeah, after my wife tried it the first time and realized how painful it was, she refused to do any more of it. So
all right. That's interesting. Yeah, thank you. Yeah, it's a big struggle trying to get participants, especially when you're doing a virtual internship or working virtually. Yeah.
Justin, we have a question in the chat.
Let's see if I can pull that up.
Or Harvey, if you prefer to just read it.
Okay, I can see it. Do you have any ideas for doing this at home at scale, having people help out, being shipped equipment, giving your experiments, work from home as both the investigator and subject? So yeah, carrying out this experiment, at home, like if we were trying to, ship people equipment, I think that would be kind of difficult. Sharing equipment around, the EEG caps, they are relatively inexpensive, but they're still not super cheap to send around to people. And, it would be hard to rely on people having equipment like that at home. So yeah, working from home is definitely a challenge with experiments like these that do require specialized equipment. It kind of requires a bit of lab space to do this in.
It is actually not that easy. Simply put, not everybody has the skills to put properly the EEG cap and to make it to work and to get the data. We have done in the BCI team in Microsoft Research the opposite thing. When we have the equipment installed in a lab and members of the BCI team go and one per day take turns to put the equipment and to record the data. This is how we did the data collection for one of our studies. But those are people members of the BCI team with certain skills and ability to work with the equipment. Unless the equipment is enormously simple to use, that could be a problem. More questions, dear colleagues?
Yeah, there's quite a bit to check, and making sure each of the electrodes have proper placements and getting a proper reading, like having the full contact with the scalp, and then just getting all the programs set up. Like I have 4 different programs running at the same time, collecting the data, transmitting it, processing it, presenting the stimulus. So it would be a little bit hard to set up if you're not fully experienced in it.
Okay, more questions, dear colleagues? More questions once, more questions two. Okay, I say we thank Justin for his talk. Thank you, Justin.
Thank you. Thank you all.
And thank you everybody for coming to this meeting. If there are no more questions, I would say this talk is over. Thank you again for coming to the final talk of Justin Kellmark's intern in Audio and Acoustics Research Group. Let me stop the recording. And how to do that?